if there are no other questions, we would go to the second talk. It's from Dr. Natalia Vtyurina from Groningen Research Institute, and she will talk about live cell fluorescence imaging platform to characterize internalization of nanomedicines by human cells.
So we have to dim the light a little bit. My talk would be a bit different from the clinical applications and the rest of the talks, but still I hope that I can entertain you with some movies. That's why we have to be a little bit in the darkness. Yeah, my name is Natalia Vtyurina. I'm a postdoctoral fellow in the group of Anna Salvati, and I'm happy to be here to present my work where I try to establish the live fluorescence imaging platform in order to characterize internalization of nanomedicines by human cells. So as we've heard through many talks on this conference, that the potential of the nanomedicine for targeted drug delivery is huge. And if you inject the nanoparticles that with a low added inside medicine, it can circulate through the whole system of the body and capable to end up in a certain organ and to cure the disease associated with this organ. But what is more important that the size of the nanoparticles is such that it's capable of interacting with the cellular machinery. And what is more important to mention that the nanoparticles can be internalized using active cellular pathways. That means we can control these processes as opposed to many small drugs that just partition through the membrane. Because of that, we are capable actually of delivering the medicine inside of the cell in the specific place at the cellular level and cure the diseases at this level. But what is the problem? That we don't know the correlation between the design of the nanoparticles, so what should be the shape, the material, or what kind of proteins, ligands, receptors should be attached to these nanoparticles in order to achieve this controlled targeted drug delivery. So these mechanisms are really poorly understood. These are fundamental mechanisms that we don't know how to design such a smart particles that will do this job. So let's go back and see what do we know about how nanoparticles go inside of the cell. And we know that there are some mechanisms that are very, very well established. And for example, this micropinocytosis, when the huge arm or membrane ruffle forms and engulfs the nanoparticle. Then glycerin-mediated endocytosis, when the vehicle is forming, that is coated with the glycerin. And there is independent from the glycerin mechanisms that are less understood. So one of the examples will be caveolin that is involved, but there are many, many other proteins that are involved, and there are many other mechanisms that could be used, but they are really not characterized. And the things even become more complex when we see that the same particle with the same size, same material, everything is the same, but it can use multiple pathways in order to be internalized. So if you change the cell line and if you change the particles, the picture completely changes. The other challenge is that most of the nanomedicines, they actually, after they've been internalized, they are trafficked to the lysosomes over here, and they're being degraded. So the goal of my project is to develop universal microscopy-based live cell fluorescence imaging platform, so that we will be able to characterize and identify involved in the uptake proteins, and we could screen the specific particles of your interest, that you designed in the cell line that you want to study. So the experimental procedure is pretty simple. So I seed the cells, I express fluorescent proteins of my interest, I expose to the nanoparticles, I go to the microscope, I record images, and then I quantify my results with the colocalization analysis. So with this movie, I would like to show you how the cell exposed to the nanoparticles look like. So you see particles diffusing outside of the cell. There are some that attach to the membrane, and some then being internalized. And if I track it for 5 hours, for example, here I show you the example how cells engulf the particles that are located on the surface of the glass slide. So it's really impressive. So you could track really at this level how they go inside and how they trafficked further. With this movie, I show you how the particles actually that just being internalized is located inside of the vehicle that is formed from the membrane proteins, and now it's released from this vehicle. Another scenario, as I mentioned, is a micropinocytosis. So here you see the membrane ruffle in red forming and actually engulfing all the nanoparticles that just appear to be nearby. But as I said, so these are just two scenarios that I showed you that are very common and very well understood, but there are many, many, many others and many, many other proteins that we should screen in order to reveal if they are involved in uptake or not. So with this movie, I show you the cell that is exposed to the nanoparticles for a few hours. In the red is a clustering, and you see that some particles are inside the vehicle formed from clustering, some are just trafficked, and you see some yellow color, meaning that these two probes are overlapping, that meaning that physically they interact between each other. So I can say that if I see this, that there is involvement of these proteins in the trafficking or in internalization, depending on the time point where I see that. So how do I quantify that? So I basically look for the overlap between two probes, and I try to take Z-stacks over time so that I can capture the whole volume of the cell. And for example, I know that my nanoparticles, after several hours, they will end up in the lysosomes. So you see a lot of yellow color here, meaning the nanoparticles are inside lysosomes. And how do you quantify that? So I take the Z-stacks. Here you see the raw image, so a section of the Z-stack, and here is a segmented binary image. So I just simply remove basically the background by doing this and take into account only the objects which intensity is high, so that's only these objects I will take further for the analysis. I do the same for the nanoparticles channel, and then I overlap these two images, and you see everything what is in yellow. That means that these particles are in the lysosomes. And then I'm able to, basically here I show the overlap regions. I'm able to quantify how many actually nanoparticles objects appear to be in the lysosomes. And for example, for this image is 36%. So I really can tell you the number of, after a certain time point, how many are there. And if I take the Z-stacks every 15 minutes, I'm able to judge about the kinetics of the nanoparticles. So here you see nanoparticles objects detected over time, and here the lysosomes object detected over time. And what is more importantly, I can quantify the number of colocalized objects over time and the fraction of colocalized nanoparticles that are in the lysosomes. And you see that every 15 minutes, if I record it, I see that nanoparticles steadily arrive to the lysosomes, and the amount of the nanoparticles there steadily increases. So this is, in my opinion, it's very impressive. So how in detail we can track such processes. And here I just show you one example with lysosomes. It was quite difficult. Took me a lot of time to be able to record such a beautiful movies that I showed you and to adjust all the parameters. Now my platform is ready, and I already have a data for 2 different cell lines, and I have a data for 5 different proteins of my interest. That I hope I'll be able to present you in the next meeting and give you an impression of how can we compare such things. And with this beautiful movie of RAP5 vehicle and nanoparticles wiggling around, I would like to end my presentation and again emphasize that if we want to achieve the active targeted drug delivery, it's very important to focus on the fundamental mechanisms that are involved in the uptake of the nanoparticles, because only that will allow us to have these processes controlled and to increase the efficiency of this process and minimizing the side effects from the nanomedicine. With that, I would like to thank my group in Groningen for their support, and thank you for your attention. I'm ready to take the questions.
So thank you very much for this really interesting talk. Are there any questions? Yeah.
Thanks very much. Very nice presentation, excellent movies. Just a couple of questions to clarify. In that particular movie, what were the particles? Polystyrene?
Yeah, this is a polystyrene fluorescent nanoparticles.
And the cell type?
This one, HEC.
Okay, so remind me, are they phagocyting or epithelial? What's the origin of those cell lines?
It's a human kidney embryon.
Ah, so it's not specialized phagocytes, no?
No.
Okay. No. So the question is then, okay, if you culture long enough, it's excellent to see the penetration inside the cell. Do you ever see exocytosis on the long term?
No, with these nanoparticles, nobody's seen it, and I don't expect to see it.
And what was the longest observation term for that? So is it 24 hours, 48, or?
It depends on how often do you want to take the image. So I do every 15 minutes for 7 hours. That's how much cell actually can survive the exposure. But of course, if you do it every 1 hour, then you can do it longer. No,
that actually was what I meant. So what was the longest observation term when you can say, no, there is still no exocytosis? It was after how many days you can say no, they don't go out?
No, I didn't track, so that was not my goal to track it. And I don't look for exocytosis. I look for very initial first stage at the moment. Of course, that could be looked.
Okay, thanks. Thanks very much.
Thank you very much. Very nice talk. I was wondering how. Because you said you have like 38% of the particles can be internalized by the cells. So do you consider about like all the particles can be uptaken to the cells, or just like some of them can be uptaken and some of them just suspend in the liquid? Do you think about this?
Yeah, this number that I showed, it's a 36%, and a certain time point for the certain cell of nanoparticles objects that appear to be in lysosomes. So if I wait longer, I expect this number to increase because more nanoparticles will arrive to lysosomes, for example, because my curve didn't really reach this stage, you know. It's still increasing, and I expect that if I wait longer, it will be increasing.
Yeah. I know it will be increasing, but do you notice some particles maybe just suspend to the liquid? They may not reach the cells, because we are also count because for the cell and the nanoparticles interaction. Because we think that if you have too much liquid, and then some particles maybe just suspend in the liquid, they will not reach to the cells. So I just a little bit. Curious about this.
Yeah, or of course, not all the nanoparticles that are used to expose the cells end up inside of the cells. That's definitely the case. And in my case, since I'm looking only for the particles that internalized, these images that I showed you, I identified the particles that are inside of the cells, I really discard everything what I see outside.
Yes, I see it's very nice way. And another question is, so you just check 5 hours, right?
5-6 hours. Yeah.
Okay, so you don't say maybe I don't know if the cells don't degrade the particles, they may be released by the cells if they eat too much after like 24 hours or 2 days.
The particles degradation.
Like the cells, if the cells not degrade the particles or digest the particles, and the particles, what will the particles do? So if you do like long time, like 2 days, and I think maybe you can say others. I don't know, but I'm just a little bit
yeah, I know that after 24 hours, for example, they're still in the lysosomes. So this is the longest time I tracked. And I see on my curve, I can identify the number of nanoparticles objects versus time. So I see that how many objects I can detect over time. And if it's steady, that means it's the amount of nanoparticles doesn't change.
Okay, thank you.
Okay, so thank you very much. Unfortunately, due to time restriction, we have to go further. Maybe you can ask your question in the break.